of clarifications from chapter 12. I don't think I put these on the equation sheet. These might be useful for you to write down. Total concentration of ligands and total concentration of your protein, your receptor, whatever it's binding to, whatever these two things are, would always be given by whatever amount was bound up and whatever amount was free-floating and is not bound. Okay? And the same thing would be true on this end. Just the total ligand concentration in any solution, in your blood, anywhere, wherever it might be, it's either bound or it's not. These are our two choices. Don't make any other assumptions unless you're explicitly told to. Okay? So this could be, trying to decide whether or not the total amount of ligand concentration is so much higher than the protein concentration that you can let total be free, like let these be equal, because the amount that gets bound up is essentially irrelevant. You'll know that because I'll tell you. Don't decide that on your own. Okay? The idea that you have perfectly specific one-to-one binding such that the bound concentration of ligand would be given by the bound concentration of protein and would be equal to the complex concentration. Again, just don't make that assumption unless it says perfectly specific one-to-one binding. If it says it, this is how you know that you can use that assumption. But these equations up here would always be valid. Okay? I will do my very best to be consistent with subscript use. Okay, this might not be true on any of the problems you see today, because again, they're old and I'm just like drag and dropping them. But for anything new on the exam that I write, I will do my best to always keep subscripts there. But when in doubt, if it's not there, it means free. Okay. There was a question that appeared on Piazza about p-total and scattered analysis. And was this just the protein of interest, or did that represent the total mixture of protein in there? How do I interpret this? Okay. This is a great question. It depends on the nature and the details of the problem. So theoretically, you want, when you're running some binding assay and you're using scattered analysis to interpret it all in the context of ligand concentration, you don't know how much protein you have. You isolated it from some cell or some tissue. You have some unknown mixture. Maybe you tried to purify it to get just what you want, but you don't know for sure if you have that. Ideally, p-total represents you only got binding to what you care about. But what that p-total actually really represents is the total amount of protein concentration, like it's related to what was actually happening in there and whether the ligand was binding it or not. And so the ligand could bind to things we don't want it to. It also could bind to the walls of our containers and stuff. And so what you can't maybe go on with p-total and say is that this concentration is exclusively the protein of interest if it came from a mix. It might not bind to everything in the mixture. You don't know for sure. This is why you would try to use some sort of non-specific subtraction so that you could maybe, if you get a p-total, you could remove out what you don't think is relevant from that. But I will, again, do my best to write questions that sort of take the ambiguity out. And of course, this time, I will be sitting outside the room and you can come ask me questions. Okay? This is the equation for dissociation constant. Okay, so again, I don't think I put the free subscripts on the equation sheet. They're implied. Okay, if they're not there, it means free. Okay. So a really small or a really low KD value means we have high affinity, strong binding, and it's also for this standard state condition where we're talking about 1 mole of protein binding 1 mole of ligand. Okay, KD is a constant for us given a particular temperature, and it's related to whatever particular protein ligand pair we're talking about. Okay, you cannot talk about the fraction that would be bound at equilibrium in real life without more information about real life concentrations. Okay, and so there was a problem on homework 8 about I want 91% to be bound at equilibrium. And so there was some confusion, I think, about, okay, but then at equilibrium, shouldn't fractional saturation be 0.5 because the ligand concentration is going to be equal to the KD, and is it always 5? So I've already forgotten what that Piazza question was because I didn't read it recently. But the way to think about this is that if you're trying to design a drug, let's say, to bind to something, you could just put a ton of drug in. I mean, maybe not to the point of killing yourself, but you could deliver a really, really, really high concentration of this ligand, this drug in your blood, such that it would bind up everything. And that's because you've put in a ligand concentration way, way higher, way, way above this KD value. And so when you first put it in, there's like this time course, everything's free. And over time, it starts binding up. And when it reaches equilibrium, the amount that's bound could be, they're all saturated because we raised that concentration high enough. So I think maybe a nice way to think of dissociation constant is sort of closer to our KEQ, our standard state, whereas real life conditions are more like the Q, our reaction quotient. Everything's not 1 to 1 mole, if that's maybe helpful. It's not a perfect analogy, but KD is sort of this relative measure for us for this binding pair in sort of a standard state imaginary scenario. How do these two things bind to each other? Then let's go over here and talk about real life concentrations of protein and ligand and whatever it is. Then we can think about how much fraction would be bound at equilibrium. KD wouldn't be changing. Okay. Any questions about that? Okay. This is a slightly new and improved figure of equilibrium dialysis. Okay, so this was you have a membrane where protein can't get through, but your ligand molecules can. And at T0, you have them separated. And when you wait for equilibrium, so the ligand molecules based on concentration difference want to flow through the dialysis membrane. Some of them are going to be able to get bound by the protein. And then additionally, they're going to keep flowing even after they start binding in order to make the free concentration on both sides also equal. Because otherwise you would have a continuous, it's going to flow till it stops based on concentration difference, like free concentration difference. So this is a nice figure that if you literally count the dots, the number of dots of ligand don't change on either side, and the number that are free and unbound to the protein out here, I think it's like 13 to 13, they're also the same. Okay, so it maybe literally helps you visualize the fact that the concentration that are not bound up to a protein on either side are going to eventually become an equilibrium. These are free on either side. This one is clearly, it doesn't seem to show 1-to-1 binding because it kind of looks like 2 or 3 can bind the protein. I mean, it's a cartoon, so it's not a perfect image. But it does sort of show you the idea that maybe you'd, if you have a concentration high enough, sufficiently above the KD of whatever the interaction is between this one ligand and this one protein, you'd like hit up all the protein and then more ligand would keep flowing until you get a free concentration even on both sides. Now, if the ligand concentration isn't high enough over here, some of the protein might get bound, but not all of it, and then you could have a fraction that is something less than 1. Okay? This is the figure I showed before. This is what it would look like in a beaker, in a tube, in a lab for real. And there was some questions on homework 8 about getting out of molar. Everything you were given was molar. There was a molar concentration inside and outside and free and bound. And how do I get into just moles? And the answer is you couldn't, given the information I gave you, because I didn't give you volumes. So clearly the volume in this tube is not the same as the volume outside of the tube. So you couldn't get into moles with the information I gave you. What you had to trust me on is I'm telling you I have some ability to measure concentration inside, outside. I know what volume I put in the beaker. I know what volume I put in my tube because I measured it before I did it. And so I can get a molar concentration. Okay? So you had no need to get out of molar, leave everybody in molar, everything's fine. The volumes might be the same. They don't have to. Doesn't matter. What is a way if I wanted to take a sample of this like liquid and I wanted to figure out what the concentration was? What are some actual analysis techniques we talked about for that? Yes. Great. I can hit my sample with some light, and I can look at a change, and I can measure absorbance, transmittance. I could tag my molecules with something radioactive. I could tag my molecules with something fluorescent, and then I have some standard curve I've already set up. I already had to preset a standard curve where I knew at this concentration, my light spec or my fluorescent spec or whatever it is looks like this. At this concentration, it's this level. At this concentration, it's this level. Then I go run my experiment. I wait for equilibrium. I pull out my sample, hit it with light, do whatever, go match it to my standard curve and say, okay, this is how much is left outside after I waited for it to bind up. Okay. Anything about chapter 12 clarifications?